Okay, so we're going to be talking about chapter 21 today, and that is lymphatic system. Lymphatic system is going to be one of our shortest chapters that we have here, so I won't keep you today for very long, and then you'll have the rest of it recorded, so I'll post that for you a little later, either today or tomorrow. And so let's go ahead and jump into the Nearpod. If you haven't joined the Nearpod yet, here, let me put the code in the chat here. And if you're watching from home, then you have a link on your Blackboard where you can run the Nearpod for the activities on your own. So let's go ahead and share the Nearpod screen with you. Or my iPad screen is what I'm going to be sharing. Okay. All right. So lymphatic system is a system that is closely associated with, hold on, let me move stuff around, but it's closely associated with immune system. So this is going to be a very nice introduction into the immune system. So a lot of these things are going to come back to us when we talk about immune system or chapter 21. Lymphatic system is going to be composed of lymphatic vessels, lymphatic structures, and then fluid that we know as lymph. This fluid, lymph, comes from the interstitial fluid, and it's going to move into blind or closed-ended lymphatic capillaries by passive transport. And if you remember when we talked about capillary exchange, I drew in a lymphatic capillary into that picture towards the end. And so we said that about 10 to 15% of fluid that's in the interstitial space gets absorbed by the lymphatic system or lymphatic capillaries. On this image, what you're looking at, you can see those lymphatic vessels distributed throughout the body. And then you can also see two tables here. You see primary lymphatic structures and secondary lymphatic structures. So we'll be dividing the lymphatic structures into primary and secondary based on their function. One thing to keep in mind is that there are only two primary lymphatic structures, and these are thymus and red bone marrow. The other lymphatic structures are going to be secondary lymphatic structures. You also see some other things shown here or labeled here, and that's the couple of ducts that we have here, and then trunks. And then at the bottom of this image that got cut off here, but you can see it in your textbook, is a lymphatic capillary, and we'll have a picture of a zoomed in lymphatic capillary here. Some other structures, so for the secondary lymphatic structures, we'll be talking about the tonsils, lymph nodes, so your spleen, and then MALT, which is mucosa-associated lymphatic tissue, okay? So that's that. So let's talk about that lymph a little more. Make sure that you remember, sorry, I don't know how to turn off that stuff from my screen. But make sure that you remember that lymphatic capillaries are going to be blind or closed-ended. What that means is that they're going to have a definite end. They're not all interconnected like what we see with the blood capillaries, but they have an end, kind of like a branch on a tree, if you want to think about it that way. So this fluid, this lymph, is going to be exactly the same by composition as your interstitial fluid, okay? And so make sure that you, if I ask you, is lymph the same as the interstitial fluid? By composition, they are identical. Okay, so whatever you can find in the interstitial fluid, when it moves into the lymphatic capillary, now we call it lymph, but there's no filtration going on. We're not losing or filtering anything out from the interstitial space. Okay, so know that by composition, they're the same. The only difference is the location. So lymph is in the lymphatic capillary or lymphatic vessel, and then interstitial fluid is in the interstitial space. Okay, so make sure that you remember that. Some things that we can find in the lymph would be water, definitely, that's the base of it. We're going to see dissolved material. There's going to be foreign materials that might be present in that tissue, cancer cells, as well as some proteins that might enter the lymphatic capillary. Ultimately, the lymphatic capillaries will merge into larger vessels, larger lymphatic vessels, and then those larger lymphatic vessels will merge into lymphatic trunks, and then ultimately they will join into duct, one of the two ducts that we have in our system. So let's look at those lymphatic capillaries a little closer. Let me switch this, but before we do that, we're gonna do a little activity here. Okay, so let's do this one. Okay, and we have what? There's 4 of us here. Oh, I guess me too here. I'll be a fox. All right, so we've got 3. I mean, if you hit the start button, if you're playing, and let's start with sound. And as always, I'm not going to play, so you pick the right answer here. Where's my chat button? I just realized I forgot to hide your names, but oops. All right, good job. Look at you guys. So awesome. Yay. All right, so let's go ahead and go on to the next slide. Okay, so I promised you, oh, well, you won today, yay. Okay, so I promised you that we would look at the lymphatic capillaries a little closer. So here you can see the lymphatic capillary enlarged. And so notice how I said that the lymphatic capillaries are blind-ended or closed-ended. So you see the ends of them, right? And that basically that's a beginning of the lymphatic capillary. They're closed-ended or blind-ended. Sometimes people get thrown off by the terminology blind-ended. And it just means that there is an end to it, or kind of like a beginning. They're not all interconnected. Like when you look at the blood capillaries, there's, it's all like a network. So anyway, so that's what blind-ended or closed-ended means. The fluid moves into the lymphatic capillaries by the fact that, or due to the fact that from the blood capillary, as the fluid filters out into the interstitial space, you're adding fluid into this space, right? So when that happens, this increases the pressure in the area a little bit. And that slight increase in hydrostatic pressure in that area pushes the fluid into the lymphatic capillaries. So that's kind of how the movement goes. When you look at the part B, the image B on your picture, you can see that the cells of the lymphatic capillary are arranged a little differently than what we saw in the blood capillary. So these cells of the lymphatic capillary are overlapping. So they're not like this next to each other, but like this. And so the pressure in the lymphatic capillaries is lower than blood. Yes, I don't have the number for that, but it would be, because you can only move the fluid down the pressure gradient. You can move it where it would be, yeah. So it has to be lower. So as the pressure increases of the fluid in that area, it pushes those overlapping cells kind of open, and fluid can go inside these capillaries, as you can see in that picture, or the B picture, the lymphatic capillary one. You can also see that lymphatic capillaries do not have basement membrane surrounding them. So that might not be something that completely jumps out in your face right away, but there's no basement membrane on the outside, surrounding these cells that make up the lymphatic capillary. And so for that reason, there has to be some other mechanism that keeps the whole structure together. And so we have these, what we call anchoring filaments. Those are those little purple lines that you see there, and they anchor the cells of the lymphatic capillary to the surrounding cells in the area. We can find the lymphatic capillaries, I forgot to mention that, but we can find lymphatic capillaries in next to the capillary beds, because their function is to absorb some extra fluid from the capillary exchange. So you would see them near capillary beds. There are some exceptions where we do not see lymphatic capillaries, and these would be in the red bone marrow, central nervous system, as well as avascular tissue. So if it's avascular, you're not going to find these lymphatic capillaries there. So I mentioned the anchoring filaments, so make sure that you, again, remember blind-ended or closed-ended vessels is what these are, okay? And it's the increase in hydrostatic pressure that pushes some of that fluid into the lymphatic capillaries. So these lymphatic capillaries are going to, as I mentioned, merge into larger vessels, and then larger vessels merge into trunks, and then the trunks merge into ducts. So let's look at the next image right here. And this one has the trunks and ducts labeled for you. These are left and right for most of them. Okay, so we have right jugular trunk, you would have left jugular trunk as well. I'll just talk about them kind of in general as far as what they drain. The jugular trunks that you see here, these are going to be draining your head and neck. Okay, so think about where your jugular is. Okay, jugular trunk is going to be nearby. So if you know your vessels, these are kind of, they should help you decide where these are. Subclavian trunk, as we move down, again, you would have left and right for these. Subclavian trunk is going to drain your upper limbs, your superficial thoracic wall, as well as the breast area. The bronchomediastinal trunk, again, left and right here. This is going to drain the deep thoracic structures. And when I say drain, I mean it receives lymph from this area. So that's what that terminology means. Then we have intestinal trunk. There's no left and right for that one. It's just intestinal trunk. And this one is going to drain or receive lymph from your structures within the abdominal area, abdominal cavity. And then lumbar trunk is the bottom one there. And lumbar trunks, left and right, are going to be receiving lymph from the pelvic organs, from your lower limbs, as well as abdominal pelvic wall. So that's how that works. These trunks will then merge into one of two ducts. And as you notice on here, there are only two ducts that we can see here, and these include your right lymphatic duct and then thoracic duct. The right lymphatic duct, as you can see on the little green man down there on that picture, right lymphatic duct is only going to drain the upper right quadrant of your body. So if you can remember that, everything else is going to be drained by the thoracic duct. And so that's that. One thing I didn't mention about the lymphatic vessels in general is that they have valves, they resemble veins. So lymphatic vessels in general are going to look very, very similar to your veins. They're going to have all those three tunics that we saw in the veins. They're going to have a large lumen, and they also will have valves. Because remember, the function of the valves in blood, in veins, was to prevent backflow because the pressure was already so low there. Well, the lymphatic system doesn't even have a pump at all. So the only thing that the lymphatic vessels rely on to move the lymph through is going to be the skeletal muscle pump, just like veins, and then the thoracic or the respiratory pump as well, just like with the veins. So these ducts, make sure that you can tell me what is drained by which duct. I will definitely have a question on the exam like that, you know, like, okay, the left arm is drained by which duct? And you would be like, oh, it's thoracic duct, right? And so know what is drained by what for sure, for as far as the ducts go. And just in general, know those trunks as well, okay? There's one structure that we see at the base of the thoracic duct, and this structure is known as the cisterna chyli. It's not chili, it's chyli. And this is an area, it's kind of, cisterna means like a cistern or like a container. And so this is an area that receives lipid-rich lymph from your digestive system. When we look at digestive system, we will see that the way that our body absorbs lipids is going to be by the means of the lymphatic system. And it has to do with the fact how our digestive system packages lipids. It creates these protein capsules that are filled with lipid. These are known as your chylomicrons. And so chylomicrons has to do with lipids. Cisterna chyli, you know, receives these chylomicrons. And then, you know, all this lymph as it goes through the thoracic duct. If you notice where the thoracic and the right lymphatic duct end, they're going to connect to the junction of the subclavian vein and the jugular vein. So where there's the subclavian and jugular meet, the duct is going to meet there or connect there as well. And so all this lymph, as it flows through the lymphatic system, it is going to then enter the blood again. It's basically what we're seeing is some of the fluid from the capillary exchange, instead of going directly into the blood, it just takes detour through the lymphatic system, and then it ends up back in the blood. So you're not keeping it in the lymphatic system forever. It's just going a little different route. And this is going to be important because we will see on those, among those vessels on this image, you see some nodules there. And these are your lymph nodes. And so as the lymph flows through the lymphatic system, it will flow through lymph nodes, and the lymph nodes will scan this fluid for any traces of infection, any pathogens, anything that should not be in your body or that your body needs to fight against. Okay, so that's the whole point of lymphatic system. Okay, we're scanning our body for pathogens. So let's look at our tissues, but I think I have a matching activity here. Okay, so let's go ahead and start this. And you're just gonna match some things together. And it's okay. I mean, it actually doesn't let you make wrong matches. If it's wrong, you can correct yourself. So on yours, I mean, I'm not gonna match it so that you're not, you just see all those choices. But on your screen, where you're logged into Nearpod, you can go ahead and start matching these. All right, good. All right, last 30 seconds. Okay, good. Almost done here. 15 seconds. You can do it. All right, so this is, again, I'll post this so you can practice these some more. It would be a kind of a nice matching activity there for you. So, okay, so let's, yes, so yeah, you will have access to this. So I'll post the student-paced or self-paced version of this. All right, so let's look at our lymphatic structures and lymphatic tissues and organs. So we can, as I've mentioned earlier, we can divide structures into, based on their function, into primary lymphatic structures and secondary lymphatic structures. Now, I didn't tell you what the criteria for the division was. And what it is their function. So your primary lymphatic structures are responsible for formation and maturation of lymphocytes. So write that down somewhere. Formation and maturation, this is going to be your primary lymphatic structures. And there are only two lymphatic structures that are primary, that are responsible for formation and maturation. And these include your red bone marrow and the thymus. Red bone marrow, you already know, that is the site of formation of formed elements. Lymphocytes or white blood cells in general, leukocytes, are a formed element. So red bone marrow is a site of formation. It's also a site of maturation of several different cells. And so that's what bone marrow does. Okay, so it's a site of hemopoiesis. The thymus, the second structure, this one is going to be responsible for maturation of T lymphocytes. And we will actually look much closer at that process in chapter 22. So I'll have thymus in a little bit. So there's not really a whole lot more that I want to say about the red bone marrow. Just know that's where all of the formed elements are formed, and that most of formed elements are also going to be either completely mature or almost fully matured. The second primary lymphatic structure includes thymus, and we can see our thymus right here. You have actually a comparison of a child's thymus versus an adult's thymus, and you can see that a thymus of a child is much larger than the thymus of an adult. And this is because during childhood, and this is going to be before puberty, our bodies are still getting to know a lot of pathogens, and so our immune system is still developing. When puberty begins, this is where the thymus goes through a process that we know as involution. And this means that it's going to get smaller in size, but also internally it's going through changes. There's a loss of thymic tissue, and some of this thymic tissue is replaced by adipose tissue. So when you look at the microscope slides of the child's thymus versus an adult's thymus, you see that the child's thymus is very purple. Okay, all that purple stuff is going to be the thymic tissue. On the adult's thymus, you see just a little bit of purple, and then the rest of it is kind of whitish, and that's the adipose tissue. Yeah, so that's that. So it's not as, our thymus is not really as active after puberty, because the idea is that we have already encountered a lot of different pathogens by that point. Okay, so that's what happens to our thymus. It shrinks in size. It also gets, a lot of that tissue gets replaced by adipose tissue. Thymus is a bilobed organ, so you can see it has two large lobes, and then within each lobe you have lobules. These lobules are, all these lobules are going to be divided into cortex and medulla parts, so each lobule has a cortex and each lobule has a medulla. The lighter parts of the lobules are going to be the medulla, the middle area. The outer edges are the darker parts, that's the cortex. You can see the lobules really well outlined in the adult's thymus. On the child's thymus, these lobules are really, really close to each other. You almost kind of lose the complete lobule site there. But you can see, definitely, you can see the dark and the light area there. So the light areas are the medulla of each lobule. The dark areas are the cortex. So it's not the whole organ that's divided into cortex and medulla. It's each lobule has a cortex and medulla. These areas are going to have different function when we talk about the maturation of T lymphocytes. So thymus is a site where, or a place where the T lymphocytes mature. Okay, so thymus is a site for T lymphocytes maturation. You can remember T lymphocytes, thymus, T, yeah, it has that letter T. And so that's what happens there. And again, we will talk about the whole process of the maturation of T lymphocytes in chapter 22. So you'll see what happens in the cortex and then what happens in the medulla. A little spoiler alert here. If you are a T lymphocyte and you travel to thymus, you have about 2% chance of surviving this process. So thymus doesn't play around. It is very important in selecting only the T lymphocytes that are actually able to do their job and do that job correctly. And it only turns out that only 2% of thymocytes that your red bone marrow produces are able to do this. So 98% of T lymphocytes or pre-T lymphocytes will die when they enter thymus. Yeah, very picky, but that's good. It's important. So that's that. That's that for thymus. I don't really have much more to say about thymus for now. Again, we'll revisit it later. So let's look at those secondary structures. So we said primary lymphatic structures, their function is formation and maturation of lymphocytes. Secondary lymphatic structures are going to house these lymphocytes. That's where most of lymphocytes like to hang out. If you remember when we looked at blood, composition of blood, you know that leukocytes as a group, white blood cells, were in that small thin band that makes up about 1% or less of your whole blood. And that's where you find your lymphocytes and all the other white blood cells. So you see that there's not a whole lot of them are in the blood. That's because they really just use the blood to transport themselves between different areas of the body. The majority of these white blood cells is going to be in these secondary lymphatic structures. So our secondary lymphatic structures are going to be divided into lymphatic organs and lymphatic nodules, or lymphatic tissues sometimes is the term that's used for that. Lymphatic organs will have a complete capsule, and lymphatic nodules or lymphatic tissues are groups of these lymphatic cells, but they are not surrounded by a capsule. Okay, so lymphatic organs have a complete capsule. The lymphatic organs include spleen and lymph nodes. And the lymphatic nodules would be your tonsils, your mold, which some people consider part of, or tonsils being part of mold. And then we'll see a lot of diffuse lymphatic nodules. And they're going to look very much identical to what you see inside of a lymph node, those diffuse lymphatic nodules. So let's start with the lymphatic organs. We'll start with the lymph nodes. Lymph nodes, let me switch here to the next slide. This is a diagram of a lymph node, and at the bottom you have a micrograph of a lymph node here as well. So lymph nodes are divided into cortex and medulla. They, as a whole node, spleen and lymph nodes. So for our lymphatic organs, spleen and lymph nodes are lymphatic organs. Everything else for the secondary thing is going to be lymphatic nodules or lymphatic tissues. The lymph nodes can vary greatly in size. They can be as small as 1 millimeter, but they can be as large as an inch. So they're varying in size, and they typically occur in clusters in our body. So if you recall that first figure that we looked at with the whole body, and you saw the little nodules, you saw that like different areas had several of them in the region. And this is important because you are basically more likely to find a leukocyte or lymphocyte that is able to recognize a pathogen if you run the whole fluid, you know, all of that lymph through several lymph nodes. So don't put your eggs in one basket and just run the lymph through one lymph node. You're going to run it through several in that area. So they're all kind of interconnected like this. You can see that the lymph is brought into the lymph node through vessels that we know as the afferent vessels. And so those are the ones that are bringing lymph to the lymph node. And then at the hilum of the lymph node, you typically find one single efferent lymphatic vessel. Sometimes there is a couple, but usually there is just one efferent lymphatic vessel that takes the filtered lymph out. And then there would be another lymph node somewhere down the line really close by, so we can scan this again or monitor this again. The different areas in our body, or the lymph nodes in different areas of our body have different names. So for example, like in the armpit area, you would have the axillary lymph node, you'd have the inguinal lymph nodes in the inguinal region. So one place where people get confused is the cervical lymph nodes. And cervical lymph nodes are in your neck. And then you also have submandibular lymph nodes, and that would be under your mandible. So make sure you keep those straight. I think they might be labeled somewhere in your textbook. So if you come across those, cervical lymph nodes, neck area, they're not in your pelvic area anywhere. Okay, we're not talking about cervix of the uterus. Here we're talking about these lymph nodes right here. So that's kind of the general description of the lymph node here. Let's look a little closer at the cortex and the medulla. So what do we see here on this lymph node? You can see that along the cortex area of a lymph node, that's the purplish area right there, you have these lymphatic nodules. Okay, so the purple part is the lymphatic nodule. Keep that in mind, because when we look at diffuse lymphatic nodules in other areas, they're going to have the same structure. They're going to look like you took it out of the lymph node and just placed it somewhere in your body. Okay, so lymphatic nodules are the same in the lymph node as they are when they're on their own. Let's look at the cortex here. So if you look at the top part of this image right here, you see the cortical sinus. So within or between these lymph nodes, lymphatic nodules, you have sinuses that the lymph flows through, okay? And these are called cortical sinuses if it's in the cortex. And the lymph will flow around. And then you can see the mantle zone. That's the darker area of the lymphatic nodule. And the mantle zone is going to be full of T cells, macrophages, and dendritic cells. These cells like to trap pathogens. So let's say that the lymph that's flowing through has something that is foreign to your body. The dendritic cells are cells that they look like they have dendrites, and they can grab any pathogens that are in there. Macrophages are going to engulf or eat any pathogens that should not be there. And then T cells are waiting there to see the message from these other cells. And we'll elaborate on that a little later. Again, would be in chapter 22. T cells are actually not able to see the pathogen directly. They have to get a message from either a dendritic cell or B lymphocyte or macrophage. And so that's why it makes sense that you would see them near each other. Okay, dendritic cells and macrophages would eat the pathogens, and then they would show it to T lymphocyte. When T lymphocyte sees it, then it can send a message. If it gets activated, it can send a message to the germinal center of the lymphatic nodule. And this area is full of B lymphocytes. And when the B lymphocytes get the message from the T lymphocytes, then they can get activated. And this is why it's called germinal center. These B lymphocytes, once they're activated, they start multiplying, and they start producing plasma cells and produce antibodies against that specific pathogen there. So that's our mantle zone and germinal center for the cortex. Now, as the lymph flows through the cortex, eventually it makes its way into the medulla, into the middle of the lymph node. And the medulla is composed of space, which is medullary sinus, and then the medullary cords. And medullary cords are simply connective tissue with B cells and T cells and macrophages kind of attached to this connective tissue or within this connective tissue. And so as the lymph flows through, again, these cells that are there can detect pathogens and eliminate it and initiate an immune response to that specific pathogen. So that's kind of how our lymph nodes work. Let's look at our spleen now. And I actually added a video to this. I embedded it. We're not going to watch it here, but it's by this, I think he's a YouTuber. He has a lot of videos, and I'm actually going to use it for your respiratory system as well. But he talks about stuff and he draws it as he talks about it. And I think he does an excellent job talking about the spleen. Like it's looking at the spleen a little more in detail than our textbook gives us. Now, you don't need to know all that extra detail, but I think it might be enriching for you to really fully understand how this spleen works. So it will be in this, it's after this slide, so it's going to be within the Nearpod. So let's talk about the spleen just in general here. Spleen is our largest lymphatic organ. So if I have some organs or some structures and I ask you which one is not a lymphatic structure, don't be picking spleen. Somewhere, and I don't know, I'm not sure that I have this on the test, but I used to have it on the lab quiz, and I would have a bunch of structures. For the duct, yes, but for the organ, the spleen is the largest organ. So people would always pick spleen as not belonging to lymphatic system. Spleen is lymphatic. It's our largest lymphatic organ. And it's an organ because it has a complete capsule. Spleen is supplied by a splenic artery. So splenic artery brings blood to the spleen. Splenic vein would take blood out of the spleen. And there is no cortex and medulla for the spleen. Okay, so spleen does not have cortex and medulla, but instead it's divided into red pulp and white pulp. The red pulp is going to contain a lot of platelets. It's actually a third of your platelets in your body are in the red pulp. And this is also where we are going to be recycling erythrocytes. So on that video that I mentioned, if you look in the second part of that video, he really explains how the erythrocytes are leaving your central artery. They're going to enter the tissue of the red pulp, the area of the red pulp, and then macrophages break these erythrocytes out. If they're the healthy erythrocytes actually do leave from the blood as well, but they are able, because they're healthy, they're able to enter the sinuses of that are collecting the blood from the spleen. But the old worn out erythrocytes, because they're now like different shapes and all that, they're not able to pass through those openings. So watch that video, and he draws it nicely, like how these cells are moving or not moving if they're sick. The white pulp is the area on the image that you see. That's kind of the lighter color. But on the microscope or the micrograph of the spleen, the white pulp is the dark edge and the lighter center of it. So that whole kind of the round or oval structure that is considered white pulp. About 25% of the whole spleen is white pulp. Majority of the spleen, about 75%, is red pulp. So white pulp, when we talk about white pulp, it's important to talk about the blood vessels. So as the splenic artery enters the spleen, it branches into smaller and smaller arteries. And eventually you will have a branch that is going to enter the white pulp, which is known as the central artery. And then there will be some branches off of that as well. And these are end arteries. So they don't connect to another vein, but rather the blood from these arteries is going to enter the sinuses that you see that are surrounding the white pulp. There you would see some B cells, T cells, and macrophages in the white pulp that would be responsible for trapping pathogens, trapping viruses, or anything like that, and such. Okay, and then in the red pulp, as I've mentioned, you're going to see recycling of the erythrocytes. And that's why it looks red, okay, because you have a lot of these erythrocytes and a lot of platelets there as well. Okay, so after it goes through the arteries, it enters the sinusoids, and then stuff gets taken out of that blood. And then from the sinusoids, the blood will be collected by venules. These venules will then merge together into larger and larger veins, and then ultimately into splenic vein that takes the blood out. And that's spleen in a nutshell. So yeah, as I said, I'm not gonna play this right now, but you can watch it on your, it's only 14 minutes, so it's not very long. Yeah, so that's that. Let's look at tonsils. Tonsils are not lymphatic organs. Tonsils are lymphatic nodules because they do not have a complete capsule. Actually, from the exposed side, they do kind of have a capsule, but underneath it, they're not completely enclosed in a capsule. So we consider them nodules. You can see on the microscope slide that you can see the lymphatic nodule, okay? And it looks like what the lymphatic nodule looked like in the lymph node. Okay, so that's kind of what I was saying. Lymphatic nodule looks the same inside of the, and it has the same structure inside of the lymph node as it does in other areas. And so in tonsils, you have these as well. And then there's a germinal center in the middle of those. We have three main tonsils. A lot of times the palatine tonsils are the ones that we understand as tonsils. The pharyngeal tonsils are also known as the adenoids. And then lingual tonsils are kind of in the posterior side of the tongue. Sometimes like people have tonsils removed because these structures, these tonsils are protecting us from things that we can inhale or ingest. So as you see, the pharyngeal tonsils are in the pharynx area, kind of at the back of the nose. The palatine tonsils, you can actually see probably your palatine tonsils unless you had them removed. If you open your mouth and you look in the mirror, you should be able to see them in the back there between these two arches that that we'll talk about in the respiratory system, but they should be visible. And if you have a like sore throat, then sometimes you can see that they're inflamed in there. But because they're kind of, they're like very kind of on the front lines for pathogens entering our body, sometimes they're very prone to getting infected. And people whose tonsils get infected very often actually do have them removed. Also, you can have these removed because they can, if they are large, too large, they may interfere with eating, like swallowing, or they may interfere with breathing. And so I believe the palatine tonsils are the ones that are most frequently removed. And then adenoids also. Lingual are probably the least removed, but they can obstruct breathing tract, so a respiratory tract. So sometimes these are removed as well. But I don't think it's as common as the other two. Okay. So that's our tonsils. Make sure that you are able to label which one is which. Okay, so we have pharyngeal tonsils in the pharynx, palatine in the palate, under the palate basically, and then lingual is associated with the tongue, and that's the posterior side of the tongue. So that's tonsils. The last thing for this chapter really involves MALT. This is the mucosa-associated lymphatic tissue, where you just have diffuse lymphatic nodules in different areas in the mucosa, where the mucosa is present. Because mucosa, wherever you have mucosa, this would be a great place for pathogens to enter your body. And so you do have a lot of lymphatic nodules there that are monitoring and trapping anything that is trying to get into your body through those areas. So we see them in a lot in the GI tract, the respiratory tract, the reproductive tract and urinary tract as well. In ileum, these are very prominent. You see a lot of them in the ileum. Ileum is the last part of the small intestine of our GI tract. And they actually have a special name in our ileum, and these are known as Peyer patches, P-E-Y-E-R, with a capital P, so Peyer patches, okay? So that's MALT. And that's basically chapter 21 in a nutshell. And that's where I'm going to end for today. And again, I'll give you the chapter 22 as a recording. So I'm gonna stop the recording now. And then you can ask questions if you have any about this.